So, what I wanted to talk about today is cancer proteomics and kinomics. This is the stuff we're doing, working on proteomics, and the new thing is kinomics, the special thing which are doing basically on a special thing. So anyway, why at first? What are kinases? They're enzymes which phosphorylate things. They add phosphor groups to certain things, and they make them work, basically. They're serine kinases and tyrosine kinases. Some of them, they're dual specific kinases, and they're receptor kinases and cytoplasmic kinases. We have in our organisms about 500 kinases, and it's about 2% of our genome, and they produce a lot of stuff what is our life. And if they get problems, we get a lot of cancers or some kind of other diseases. All of kinases, they are involved in a lot of things, which you can see in this little thing, receptor tyrosine kinases assemble together because they get some growth factors, then they get phosphorylated to each other, and then they phosphorylate other proteins, and it goes on, and at the end that is up to growth to the DNA, which basically makes things like apoptosis, migration, growth, and so on and so on. Otherwise, that's just the biology. Why still we are interested in biology of kinases? Because they're targets of drugs. And one of the first drugs is the thing of the RB2 receptor tyrosine kinase, which normally works as it's supposed, but in breast cancer, sometimes it could be overexpressed, and then it excuse me, but it screws the whole thing. And when these two receptors come together, they can produce signal which cause cancer. So one of the first drugs which was invented was Herceptin, which was the antibody which prevents this happening, and that's how sorry, that's how the drug works. It is a target. Another story is a glytic. That is a kinase inhibitor, which inhibits the able kinase, which normally works like an able kinase. However, when there is a problem with the able kinase, especially in leukemia, that is when there is BCR and able is getting together because of targeting each other, and able becomes an abnormal. So this gene is becoming an abnormal, and the glytic was an inhibitor, which was the first who was actually working on stopping this kinase, and 90% of this kinases and of the cancers is stopped. So why we're interested? Another thing is kinases are interesting as biomarkers, and well, that's from our work. FLT3 is a receptor tyrosine kinase, which is working sometimes against cancer, sometimes for the cancer. In our work, we showed that if patients have less FLT3, they have way better survival than the patients which have more than that. So, as I said, there are three ways of we should be interested in kinases. That's one is basic science. Another thing is because they are drug targets, and the third thing is they could be biomarkers. My talk today is going to be based mostly on, because they're the biomarkers. Otherwise, I've worked a lot as drug stuff. Anyway, so we'll start. We've done some work on how the drug, which was developed a long time ago, and we worked on the stuff that this drug really stops some proliferation and some other stuff with breast cancers, cells, and we found out that we could develop some cells which are resistant to this drug, and then we wanted to go through why it has happened, and so on. So basically, in short, in this, you can see that the major target of this drug is NHQ01, and there is some different ways how cells go just bam with stuff which don't have this drug versus when they have a resistance, then there is some the cells become kind of cancer stem cells, and they become resistant to everything, and so on. So yeah, in short, so the major thing what we used was these MIBs, so to say. Maybe it's I'm not sure if you see them too well. Anyway, these are the beads which are covered with kinase inhibitors. But in this case, the inhibitors, they're not used to inhibit kinases. They're used to capture them, because in kinase there is this pocket which binds the inhibitor. So these are very broad inhibitors. We catch all the kinases, basically all the kinases which ever exist, because there are 5 inhibitors involved in this process. So we catch all these kinases, we pull them out of a cell lysate, and we finally go through the proteomics and find out what is up, what is down, and so on. On the other hand, we did just simple phosphoproteomic, so finally finding out which proteins are phosphorylated and so on. And then basically we found a lot of things. Obviously, at the end, the thing was that we were concentrating about the things which pass. That was bioinformatics, basically. So finally, we found that a lot of those things which were up or down in our protein kinase thing, and they were phosphorylated or not, and so on and so on. We found a lot of some clusters, and we see that there is a cluster of anti-apoptotic signaling, of course, a lot of cell cycle stemness, which is I will explain that later, and there were a lot of apoptosis things. And what you see here, and what is red things were up. What is blue, things were down. And the thing is that you see that, anti-apoptotic, stemness, cell cycle, they were always up, except for this guy. Anti-apoptosis, they were always down. And finally, with all the bioinformatics and just sometimes me just looking at things, what we found that we could actually put everything into this pathway. So what you see, which things have a color, these are the things which we found, but then when you think bioinformatically and so on, that it comes into this pathway where CKIP, the receptor goes down, and then we come back to mTOR, who goes into stemness and anti-apoptotic, and JNK, who could be pro apoptotic, it goes down. Later, already with simple westerns and stuff like that, and we saw the same thing with JNK. It does do what we saw. Other thing, which is important, then at the end, what we kind of saw that the cells are going to be becoming basically these stem cancer stem cells, and we checked a lot of things which could lead to that. I mean, I don't want to talk too much, but you see that the things which it was made both with qPCR and proteomics, it was always like, the resistant cells were going up with stem kind of profile. And at the end, we saw that, if we use with these actually resistant cells, if we use any other stuff like, classical stuff, like you see it, doxatel, fluoracil, oxaplatin, and so on and so on, it always works still. It doesn't matter if these cells are resistant to our stuff. They still die as well as anything. But if we use inhibitor against this CK receptor, there is no difference at all. So they are totally resistant to anything which has to do with stemness or whatever. Another thing, what we use that is a little bit not like cancer, but I will still explain. So there is a thick so thick is bringing this disease to animals, especially to cows. And the thing is that the macrophages of these cows become totally cancerous. They are not working as normal macrophages. They are totally cancerous. That's why I got interested in this, and we worked with the same beats I just told you about. We were working on how it happened, because it's totally interesting, because this parasite is attaching to our or cows uh, basically all the DNAs and stuff like that, and it is using when cells are splitting. They are splitting as well, these parasites. So they are working through kinases, they're working through CDKs and stuff like that, just to make a cell cycle, and they make these cells total cancer cells. So yeah, we worked on that, and just in short, we found that these TEC12 cells, they are these infected cells. So we found again all these kinases, which are down regulated, because I guess this parasite didn't want that. We found some up regulated, and which are involved in special stuff. And another thing was, which is not connected to cancer, but anyway, we could also get some kinases from the organism, from this Theileria, which I think is also important, because if the cancer comes from some organism, it's nice to know that it comes from some kind of organism. It could be. Anyway, next slide is we used some protein chips, which work like this. So we in A, you see, we use some kind of label for all the proteins, any proteins, and then we use on the chip, protein chip, we use antibodies which bind only certain proteins. So in this case, at the C, you see that only proteins which are, of course, labeled, but only those bind to the chip. So we know that it's pure and stuff like that. And it was nice that actually we used that in breast cancer. So just preliminary, I can say that what we found as, yeah, in this case, a lot of phosphorylation, of course, but of course, there are interesting things like sodium transport, or like response to heat, knowing autophagy and so on. Then, when you look at the KEG, which is that pathway, is basically of course, but yeah, it shows some cancer again, and autophagy and so on, and it also, when you look about the diseases which we already know, we can see that there are already some, I mean, we can forget other diseases, but there is already ovarian cancer known, stomach cancer, liver cancer, and so on. So it's interesting to find these kind of things, and we also use the same things in this ruptured abdominal anomalies, which is not a cancer, but still it's a disease. It's our thing. With major thing, there are a lot of interesting things, which is mostly phosphorylation and so on. However, when my student worked a little bit, sorry for Lithuanian here, so when he worked a little bit on what is most important, there was mostly proliferation, apoptosis, cell inhibition, and migration of the cells, and angiogenesis. Yeah, and they also had a lot of signaling things, which, I mean, reality is that VGFR signaling, SCF and stuff like that, it's already known in biology. So we know that that stuff is involved in veins and stuff like that. Yeah, and again, what we found in this disease still comes back to that these things are involved in breast cancer, lung, skin, melanoma, carcinoma, I mean, a lot of things. So I guess that is because of time. So just thinking of some people who were involved in these things, and yeah, and I'm open to questions.
Thank you, Tom, for your presentation. Now floor is open for the questions. If anyone have. I can't able to see any further questions. Thank you, Jonas, for your wonderful presentation.